Alright class, now we move to the second major part of protein synthesis inhibitors, the 30S ribosomal subunit. In the previous lecture, we studied drugs acting on 50S. Now we will focus on drugs that act on 30S, which is the smaller subunit of the bacterial ribosome. Most important groups here are aminoglycosides, tetracyclines, and glycylcyclines. Although they all act on the same 30S subunit, their mechanisms are quite different. First, aminoglycosides cause misreading of mRNA. This means the bacteria start making wrong proteins, which are non-functional and toxic. Because of this, aminoglycosides are usually bactericidal, which is different from most protein synthesis inhibitors. On the other hand, tetracyclines and glycylcyclines work by blocking the binding of tRNA to the ribosome. So amino acids cannot be added to the growing protein chain. This stops protein synthesis, but the effect is usually bacteriostatic. In this lecture, we will also compare two important concepts: misreading versus blocking, because this difference explains why some drugs kill bacteria while others only inhibit growth. We will also discuss toxicity profiles, because drugs like aminoglycosides are very effective but can cause nephrotoxicity and ototoxicity. Finally, we will look at resistance mechanisms, which are increasingly important in clinical practice. By the end of this lecture, you should clearly understand how 30S inhibitors work and how they differ from each other. First, you should be able to differentiate the mechanisms. Aminoglycosides cause misreading of mRNA, leading to formation of abnormal proteins and bacterial death. In contrast, tetracyclines block the binding of tRNA, so amino acids cannot be added, and protein synthesis stops. Second, you should be able to identify key drugs in each class. This includes aminoglycosides, tetracyclines, and glycylcyclines, and understand their spectrum of activity against different bacteria. Third, you should understand the major adverse effects. Aminoglycosides are associated with nephrotoxicity and ototoxicity, while tetracyclines can cause phototoxicity and other effects. You should also know the importance of monitoring during therapy. Finally, you should be able to explain resistance mechanisms. These include drug-modifying enzymes in aminoglycosides, AIMs, and efflux pumps or ribosomal protection in tetracyclines. So the key idea is, understand mechanism differences, know the drugs, recognize toxicities, and connect resistance with clinical use. Alright class, now we will understand this table in a simple and logical way. This table summarizes all major 30S inhibitors, so focus on differences between the classes. First, look at aminoglycosides. These drugs act by causing misreading of mRNA at the 30S subunit. Because of this, bacteria produce abnormal and non-functional proteins, which leads to bacterial death. That is why these drugs are bactericidal, which is different from most protein synthesis inhibitors. Examples include gentamicin and amikacin. Their spectrum is mainly gram-negative bacteria, especially severe infections like sepsis. Next, we have tetracyclines. These drugs work differently. They block the binding of tRNA to the ribosome. So amino acids cannot be added to the growing protein chain. Because they only stop growth and do not kill bacteria directly, they are bacteriostatic. Examples include doxycycline and tetracycline. Their spectrum is broad, including atypical organisms, so they are commonly used in atypical infections. Finally, we have glycylcyclines, which are newer drugs related to tetracyclines. The main example is tigecycline. These drugs are designed to overcome resistance, especially efflux pumps. So even when bacteria resist tetracyclines, glycylcyclines can still work. They are also bacteriostatic, and have a very broad spectrum, including many resistant organisms. That is why they are used in multi-drug-resistant, MDR, infections. So the key idea is, aminoglycosides kill bacteria by misreading mRNA, while tetracyclines and glycylcyclines stop protein synthesis by blocking tRNA, with glycylcyclines designed to overcome resistance. Alright class, now we go deep into one of the most important groups of 30S inhibitors, aminoglycosides. These are very powerful drugs, mainly used in serious gram-negative infections. First, look at the key agents. The most commonly used drugs include gentamicin, amikacin, and tobramycin. Gentamicin is widely used for general gram-negative coverage. Tobramycin is especially useful against Pseudomonas. Amikacin is more reserved and used in multi-drug-resistant (MDR) infections. Other drugs like streptomycin are used in tuberculosis, and neomycin is used topically because it is too toxic for systemic use. Now understand the mechanism of action, which is very important. Aminoglycosides bind irreversibly to the 30S ribosomal subunit. This causes misreading of mRNA, so wrong amino acids are inserted into proteins. As a result, bacteria produce defective proteins, which damage the cell and lead to death. That is why aminoglycosides are bactericidal, unlike most protein synthesis inhibitors. Now coming to clinical pharmacology, these drugs show concentration-dependent killing. This means the higher the drug concentration, the better the bacterial killing. So we aim for a high peak level. They also show a post-antibiotic effect, PA. Even after the drug level falls, bacterial growth remains suppressed for some time. Because of this, aminoglycosides are often given as once-daily dosing, which improves efficacy and reduces toxicity. Another important concept is synergy. Aminoglycosides are often combined with beta-lactam antibiotics like ampicillin. Beta-lactams damage the cell wall, allowing aminoglycosides to enter the bacteria more easily. This combination is especially useful in conditions like endocarditis. Now we must discuss toxicity, which is very important. These drugs have a narrow therapeutic index, so monitoring is required. The most important toxicities are nephrotoxicity, right-pointing arrow damage to kidney tubules, usually reversible; ototoxicity, right-pointing arrow damage to hearing and balance, can be irreversible; neuromuscular blockade, right-pointing arrow, rare but important, especially in patients with myasthenia gravis. So the key idea is, aminoglycosides cause misreading of mRNA and kill bacteria. Right-pointing arrow, strong gram-negative activity; right-pointing arrow, concentration-dependent killing with PA; right-pointing arrow but require careful monitoring due to toxicity. Now we move to the second major group of 30S inhibitors, tetracyclines. These are very commonly used drugs, especially in outpatient settings. First, look at the classification. Tetracyclines are divided based on their duration of action. Short-acting drugs include tetracycline and oxytetracycline. Long-acting drugs include doxycycline and minocycline, which are more commonly used because they have better absorption and longer half-life. Now coming to the mechanism of action, which is very important. These drugs bind reversibly to the 30S ribosomal subunit. They block the binding of amino acid tRNA to the A site of the ribosome. So amino acids cannot be added to the growing protein chain. As a result, protein synthesis stops. Since they do not kill bacteria directly, they are bacteriostatic. Also remember, their entry into the bacterial cell is energy dependent. Now let us discuss the spectrum, which is very broad. Tetracyclines are active against gram-positive and gram-negative bacteria, but they are especially important for intracellular and atypical organisms. These include Rickettsia, Chlamydia, Mycoplasma, and Borrelia. That is why doxycycline is widely used in diseases like atypical pneumonia, Lyme disease, and rickettsial infections. Now coming to adverse effects and safety, which are very important. These drugs can bind with calcium, magnesium, and iron, leading to tooth discoloration and effects on bone growth. That is why they are contraindicated in children and pregnant women. Another important effect is photosensitivity, especially with doxycycline, so patients can develop sunburn easily. They can also cause gastrointestinal irritation, including esophagitis, so patients should take the drug with water and avoid lying down immediately. So the key idea is, tetracyclines block tRNA binding at 30S right-pointing arrow broad spectrum, especially for intracellular organisms. Right-pointing arrow, bacteriostatic right-pointing arrow, but important side effects like tooth discoloration and photosensitivity must be remembered. Alright class, now we discuss glycylcyclines, and the main drug here is tigecycline. This is a newer drug developed to overcome the limitations of tetracyclines. First, understand the mechanism and advantage. Tigecycline is structurally derived from minocycline, but it has been modified to overcome resistance. Like tetracyclines, it binds to the 30S ribosomal subunit and blocks tRNA binding, so protein synthesis is inhibited. However, it binds with higher affinity and is less affected by common resistance mechanisms. Now the key advantage is that tigecycline can overcome tetracycline resistance. Many bacteria resist tetracyclines using efflux pumps, TETA, TET-K, or ribosomal protection proteins, TET-M. Tigecycline is designed to bypass these mechanisms, so it remains effective even in resistant strains. Because of this, tigecycline has a very broad spectrum, including multi-drug-resistant organisms such as MRSA, VRE, and many gram-negative bacteria, including anaerobes. That is why it is used in serious infections like complicated intra-abdominal infections and skin and soft tissue infections. However, there is a very important limitation. Tigecycline has a large volume of distribution, meaning it distributes widely into tissues but has low levels in the blood. Because of this, it is not suitable for bloodstream infections, bacteremia or sepsis. Finally, consider adverse effects. The most common are nausea and vomiting, which can occur in a significant number of patients. So the key idea is, tigecycline blocks tRNA binding like tetracyclines but overcomes resistance right-pointing arrow very broad spectrum including MDR organisms right-pointing arrow but not used in bacteremia due to low blood levels. Alright class, now we move to a very important topic, resistance mechanisms. If you understand this slide clearly, you will understand why some antibiotics fail in clinical practice. First, focus on aminoglycosides. The most important mechanism here is drug modification by enzymes. These are called aminoglycoside modifying enzymes, AIMs. There are three main types: AAC, acetyl transferases, ANT, nucleotide L transferases, and APH, phosphotransferases. These enzymes chemically modify the drug, so it can no longer bind to the ribosome. Second, there is target modification. Bacteria can methylate the 16S rRNA using enzymes like RMT and ARMA. This prevents aminoglycosides from binding to the 30S subunit, leading to high-level resistance. Third, bacteria may show reduced drug uptake. Aminoglycosides require oxygen-dependent transport to enter the cell. So in anaerobic conditions or when membrane permeability is reduced, the drug cannot enter effectively. Now look at tetracyclines. The most common mechanism is efflux pumps. Genes like TET, A, TET, B, TET, K, TET, L, actively pump the drug out of the bacterial cell. So even if the drug enters, it is removed quickly. Second mechanism is ribosomal protection proteins. Proteins like TET, M, and TET, O, bind to the ribosome and displace tetracycline, allowing protein synthesis to continue. There is also enzymatic inactivation, such as TET-X enzymes, but this is less common and still emerging. Now focus on the center of the slide, genetic spread. These resistance genes are often located on plasmids and transposons. This allows rapid transfer between bacteria. Also remember, resistance genes for different antibiotics can be present together. For example, aminoglycoside resistance genes and ESBL genes can be on the same plasmid. This leads to multi-drug resistance. Finally, these resistant bacteria are widely found in both clinical settings and agricultural environments, which increases the spread. So the key idea is, aminoglycosides right-pointing arrow enzyme modification plus target mutation plus reduced uptake. Tetracyclines right-pointing arrow efflux pumps plus ribosomal protection. All resistance spreads through mobile genetic elements, leading to MDR organisms. Alright class, this slide explains how we dose and monitor these drugs in real patients. First, focus on aminoglycosides. These drugs show concentration-dependent killing, meaning higher peak concentration gives better bacterial killing. So our target is to achieve a high Cmax-MIC ratio, ideally around 8 to 10 times the mic. They also have a post-antibiotic effect, PA. Even when the drug level falls below mic, bacterial growth remains suppressed. Because of this, we prefer once-daily high-dose therapy. It improves efficacy and reduces toxicity. Now comes monitoring, which is very important. We measure peak level right-pointing arrow to ensure efficacy. Trough level right-pointing arrow to prevent toxicity. Trough levels must be kept low, especially with drugs like gentamicin, to avoid nephrotoxicity and ototoxicity. Now look at the graph. It shows a high peak concentration, followed by a gradual decline. Even when levels drop below mic, the PK continues, which is beneficial. Now coming to tetracyclines. The key point here is absorption. These drugs form chelates with divalent cations like calcium, magnesium, and iron. So if a patient takes them with milk, antacids, or iron supplements, absorption will decrease significantly. One exception is doxycycline, which is less affected by food. So the key idea is, aminoglycosides right pointing arrow high peak, once daily dosing, monitor levels carefully. Tetracyclines right pointing arrow watch absorption, avoid metals like Ca squared, Mg squared, Fe squared. Alright class, now let us apply everything we have learned to a clinical case. We have a 65-year-old male with a history of valve replacement. He presents with persistent fever, malaise, and a new murmur. On echocardiography, there is a vegetation on the mitral valve, so the diagnosis is infective endocarditis. Now look at the microbiology. Blood culture shows Enterococcus faecalis. One very important test here is to check for high-level aminoglycoside resistance, HLAR, because it will affect treatment choice. The AST shows that the organism is susceptible to ampicillin and vancomycin, and gentamicin is also susceptible, which is important for synergy. Now coming to the management strategy, and this is the key concept. Enterococci are not effectively killed by a single drug. They are bacteriostatic with monotherapy. So we need synergistic therapy. We combine a cell wall inhibitor, ampicillin, with an aminoglycoside, gentamicin. The beta-lactam weakens the cell wall and allows better entry of aminoglycoside, leading to bactericidal effect. Treatment duration is long, usually 4 to 6 weeks, because endocarditis involves deep-seated infection. If there is HLAR or renal issues, we cannot use gentamicin. In that case, we use double beta-lactam therapy, such as ampicillin plus ceftriaxone. Finally, monitoring is very important. We must check gentamicin levels. Peak ensures efficacy. Trough should be low to avoid nephrotoxicity. Also, sometimes surgical intervention like valve replacement may be needed if the infection persists. So the key idea is, Enterococcus right pointing arrow needs combination therapy right pointing arrow beta-lactam plus aminoglycoside for synergy right pointing arrow long treatment with careful monitoring. Alright class, now let us quickly summarize what we have learned today. First, understand the mechanisms of action. Aminoglycosides act irreversibly on the 30S subunit and cause mRNA misreading, so they are bactericidal. In contrast, tetracyclines and glycylcyclines act reversibly, blocking tRNA binding, so they are bacteriostatic. Second, look at resistance mechanisms. Aminoglycosides are mainly affected by drug-modifying enzymes, AIMs, and target modification. Tetracyclines are mainly affected by efflux pumps and ribosomal protection proteins. All of these resistance genes can spread through plasmids and transposons, leading to multi-drug resistance. Third, focus on PK/PD concepts. Aminoglycosides show concentration-dependent killing, so we aim for a high peak concentration and monitor levels carefully to avoid toxicity. Tetracyclines require attention to absorption, especially avoiding calcium, magnesium, and iron, and they should not be used in pregnancy or young children. Finally, consider their clinical importance in MDR infections. Despite resistance, drugs like amikacin and tigecycline remain important options for treating multi-drug resistant organisms and complicated infections. So the key idea is, different mechanisms right pointing arrow different resistance right pointing arrow different dosing strategies right pointing arrow all still clinically important, especially in MDR infections.